Genetic variation phenotypically presents itself in many ways, and in humans, one of the most conspicuous variations in phenotype is hair color. Look around you and notice the stark difference between light hair and dark hair. In fact, the frequencies of these hair colors is different throughout the world, with blonde hair being found most frequently in Northern Europe. Scientists have questioned why some hair is more pigmented than others, and they hypothesized that for many Northern European blondes, the answer lies in a non-coding region of DNA. David Kingsley, an evolutionary geneticist at Stanford University, found that a single nucleotide polymorphism, or SNP, was the key to the coloration of stickleback fish. Changes in the regulation of the gene led to fairer or darker skin, depending on the murkiness of the water. Kingsley's team turned to mice to determine whether that SNP was part of the regulatory DNA for human KITLG. Mice with DNA that was reversed in that region were lighter or even white instead of the usual brown, which indicated to researchers that they were on the right track. Researchers subsequently made two variations of the human version of that gene to put into mice. In one variant, they left the blonde generating SNP intact, and in the other variant, they changed that SNP to another base to resemble DNA of a brunette. They inserted just one copy of the DNA variant into each mouse. Mice, heterozygous and homozygous for the blonde generating variant, had a lighter coat than mice without that mutation. In mice, researchers concluded that a SNP in the non-coding region is responsible for dramatically affecting the regulation of the KITL gene and affecting hair pigmentation. In humans, this SNP occurs on chromosome 12 and affects the regulation of the KITLG gene. As the graph indicates, there is a strong correlation between the SNP upstream of the KITLG gene and blonde hair, depicted as the peak on the graph. When adenine is substituted by a guanine in this particular region to form the SNP, the expression of the gene reduces by 20% and less pigmentation in hair, and only hair is the result. These observations show that even a single nucleotide change in a non-coding region of DNA can result in dramatic phenotypic differences. And understanding how a regulation mechanism can affect the body allows geneticists to gain important insight as to how these regions of DNA affect susceptibility to various diseases.